Today I will introduce the paper Therapy-Resistant Tumor Microvascular Endothelial Cells Contribute to Treatment Failure in Glioblastoma Multiforme, published in Nature this year 2013. Briefly, this paper tries to show how microvascular endothelial cells in GBM support GBM stem cells growth and stemness, hence contributing to the resistance of GBM to especially radiotherapy. So in this slide, we can see that tumors are driven by a rare fraction of tumor cells named cancer stem cells. Cancer stem cells are capable of self-renewal and multipotency. In addition to these hallmarks of stemness, cancer stem cells are highly resistant to conventional therapies due to the presence of transporters, efficient drug repair systems, and etc. So, cancer stem cells are maintained by a local microenvironment of microvascular endothelial cells. This maintains the cancer stem cells in a state of stemness that is capable of self-renewal. Cancer stem cells are also capable of differentiating into endothelial-like cells. These cells form tubular structures and express some endothelial cell surface markers, but not all of them. Hence, they are not true endothelial cells. So under radiation therapy, these endothelial cells and endothelial-like glioblastoma cells mainly undergo senescence rather than cell death. In their senescent state, they're still capable of supporting tumor glioblastoma stem cells like non-senescent cells. In this slide, we can see experiments. In the experiments in this paper, first they used two glioblastoma stem cell lines, G73 and G408, taken directly from patient samples. They first tested the tumorigenicity of CD133 positive cells, and as shown in the graph on the lower right side, CD133 positive cells had more clonogenic formation than CD133 negative cells. Thus, they were able to show that they were truly working with the stem cells. And secondly, they showed that radiation truly does cause the cells to undergo senescence. Beta-galactosidase is a widely used biomarker of cell senescence and aging. Under high radiation levels, there's increased staining of beta-galactosidase, indicating that the cells underwent senescence. There was also a marked increase in P16 production, which indicates that the cells were also undergoing cellular senescence. Next, they cultured CD133 positive cells with senescent and non-senescent endothelial cells, and they observed an insignificant difference in the total number of CD133 positive cells. The CD133 positive population did not reduce, implying that even in the senescent nature, the endothelial cells were still supporting the CD133 positive cells. They also checked the radiation-induced cell death of CD133 positive cells when co-cultured with senescent and non-senescent endothelial cells. The difference in cell death percentage was less than 1% in the G73 cell line, hence further supporting that non-senescent cells were still able to support radiation resistance in glioblastoma stem cells. In the last part of the results, they showed that glioblastoma stem cells can differentiate into endothelial-like glioblastoma cells and are radiation resistant and support the expansion of glioblastoma cancer stem cells. When they stained the eGBMs with beta-galactosidase after radiation, the endothelial-like GBM cells stained blue, indicating senescence, while the parental GBM stem cells underwent cell death. Therefore, in conclusion, they were able to show that some GBM cancer stem cells differentiate into endothelial-like glioblastoma cells, and tumor microvascular endothelial cells and glioblastoma endothelial-like cells are highly resistant to radiation treatment and undergo senescence. The senescent cells are still metabolically active and still secrete factors that support the stemness and growth of the glioblastoma stem cells. In this next paper, we will talk about a new way of Overcoming Drug Resistance in Glioblastoma. The title of the paper is Melatonin-Induced Methylation of the ABCG2 BCRP promoter as a novel mechanism to overcome multidrug resistance in brain tumor cells. A brief overview. In this paper, they will be showing how melatonin, among other functions, also decreases expression of ABCG2 transporters responsible for tumor resistance of GBM to chemotherapy. To begin, brain tumor stem cells are a built-in population of multidrug-resistant pluripotent cells that can survive chemotherapy and repopulate the tumor after treatment. Therapeutic approaches need to target the tumor bulk and the brain tumor stem cells. Melatonin is an indolamine involved in the regulation of chronobiological rhythms and endocrine function. It also has anti-proliferative and cytotoxic effects. It has direct and indirect antioxidant effects. So co-administration of melatonin and chemotherapy increased effectiveness of the chemotherapy. Melatonin sensitizes glioblastoma against trail-induced cell death. Now, a newfound function is melatonin has been found to decrease expression of adenosine triphosphate cassette ABC superfamily ABCG2 BCRP responsible for tumor resistance to chemotherapeutic drugs. So on this slide, first they tested the ability of melatonin to enhance toxicity of chemotherapy on brain tumor stem cells. They tested the effect of melatonin on the toxicity of chemotherapeutic drugs with 3 brain tumor stem cell lines, NSC323, NSC72, and NSC11. They treated melatonin in combination with doxorubicin, temozolomide, and mitoxantrone for 48 hours. Then the CI values and the IC50 median effect were calculated to test toxicity. IC50 is a quantitative measure that indicates how much of a particular drug is needed to inhibit a given biological process, e.g. a cell, by half. There were decreased IC50 values when drug was combined with melatonin. This showed that melatonin greatly enhanced the cytotoxicity of the therapeutic drugs. Secondly, they showed that melatonin decreased expression of ABCG2 BCRP in brain tumor stem cells. Brain tumor stem cells showed higher mRNA and protein levels of ABCG2 BCRP and ABCB1 MDR1 than glioblastoma cell lines, indicating higher drug resistance. Melatonin-induced decrease in mRNA and protein levels of ABCG2 BCRP. Transporter activity was measured by a drug efflux assay, and there was an increase in drug intercellular content. This means that the drug was not being exported out of the cell using the ABC transporter. In this slide, melatonin regulates methylation status of ABCG2 BRCP promoter. Tumor samples had lower methylation levels, so higher expression of ABCG2. Treatment with melatonin induces hypermethylation. So DNA methyltransferase inhibitor 5, azacitidine, AZA, prevents melatonin effect on expression of ABCG2 and melatonin synergistic effect. So when cells are treated with AZA, they are more resistant to chemotherapeutic drugs. So lastly, they also observed that melatonin decreases chemoresistance in glioblastoma cell lines like A172. There was decreased IC50 values and a synergistic toxic effect observed, as well as an increased promoter methylation that could be blocked by AZA. Thus, melatonin targets both tumor bulk cells and brain tumor stem cells. So in conclusion, these results strongly indicate that melatonin can target both brain tumor stem cells and other glioblastoma cell lines. It has a synergistic effect when used with chemotherapeutic drugs. Melatonin affects expression of the exporter ABCG2. In the normal brain, ABCG2 gene is methylated, hence it has a lower expression. In GBM and other stem cells, ABCG2 is less methylated, thus it is more expressed in these cells. Melatonin induces a significant increase in methylation of ABCG2, thus it is less expressed in glioma. This leads to more drug retention and hence greater therapeutic efficacy when you use melatonin.